Okay, great. And I apologize if my mic's a bit quiet. I tried raising the input volume and I couldn't. It just kept going back to where it started. Okay, so hi, my name is Abel Vargas. I'm a third-year biochemistry student working in Dr. Catherine Borkovich's lab in the microbiology department. And my presentation is on PTH11 like GPCRs and how they regulate growth in Neurospora crassa. So I want to start off first with sort of the big picture as to why this is why we're looking into this in the first place. And part of that is biofuels. In the production of biofuels, there's like multiple points in which you can improve the output and therefore the efficiency of the whole process. The step that like we're really focused on is the breakdown of biomass. So that is the breakdown so generally cellulose into glucose and its subsequent conversion into ethanol, which can then be used for biofuels. Now, this is traditionally done by fungi such as Trichoderma rhizae and bacteria such as modified E. coli. And generally, if you have a better breakdown of this biomass, you have better efficiency, better output. So what we're really focusing on are what genes can we find that would help improve the breakdown of this biomass so we can get even more output out of what we're getting. Really, the whole reason we want to improve output is because potentially biofuels could be a part of the puzzle as to reducing our reliance on non-renewable fossil fuels and in turn help with addressing climate change. So with this, we wanted to look at an organism for studying these genes, and we're looking into Neurospora crassa. The reason that we're looking at it as a model organism for filamentous fungi is, first of all, it makes cellulases, so it can actually break down cellulose, which is pretty important when you're studying this. Additionally, its genome has already been fully sequenced. Gene deletion mutants are readily available for most of the genes. They're usually deleted and replaced with an antibiotic marker. And additionally, it grows really well under lab conditions. And once you have the genes of interest, it's very easy to modify. So I mentioned earlier that this was about GPCRs. So before I get into that, I kind of want to give a brief overview of the G protein cycle. So GPCRs are transmembrane proteins that are really important for a lot of different pathways. For us, for example, they're used in like sight and taste. So the GPCR cycle begins when a ligand binds to this GPCR, which is associated with a heterotrimeric G protein. Heterotrimeric just means it has 3 different subunits. Once that ligand binds, the GDP on the G alpha subunit is exchanged for GTP. Once that exchange occurs, G alpha subunit dissociates from the G beta gamma subunits, and they can then act on their downstream effectors. For the case of our experiment, we theorized that the downstream effectors of these GPCRs could be cellulose degradation and or carbon sensing pathways. The signal is then terminated once the GTP on the G alpha subunit is hydrolyzed for GDP. This allows the G alpha subunit to reassociate with the G beta gamma subunits, and it reassociates the GPCR, and the signal can begin once again upon the binding of another ligand. So this is generally GPCR, and we wanted to, we're looking at G protein signaling because we have previous research that shows it's necessary for cellulose degradation. This is a previous experiment done by one of our grad students in the lab, Logan Collier. This study was published in 2020. And this graph shows what happens when you delete some of these subunits for these G proteins. These refer to the G alpha subunits, as Neurospora has 3 of them. This is the G beta subunit, and these are the G gamma subunits. And as you can see by this paper, once you deleted some of these subunits, you lose all cellulase activity, which indicates that these G proteins are necessary for cellulase activity, and without them, you cannot degrade cellulose. So knowing that G protein signaling generally is important for cellulose degradation, the real question is now what GPCRs are harboring the G proteins for these pathways, because these G proteins can't act alone. They're always acting in accordance with a GPCR. So to answer that, we're focusing on PTH11 like GPCRs. PTH11 like GPCRs is the largest GPCR family in Neurospora, and it's highly conserved across this fungal family. The reason it's called PTH11 like, it's because it's related to a GPCR in Magnaportha eriziae, which is a major plant pathogen. This plant pathogen causes rice blast, which is responsible for killing enough crop to feed millions of people a year. That GPCR in question, PTH11, is actually required for that plant, for that fungus to be pathogenic. This tree off to the side shows the relationship of all the different PTH11 like GPCRs found in Neurospora crassa to each other and to the PTH11 GPCR in Magnaportha. Additionally, these little squares show the expression levels of each of the GPCRs on different carbon sources. So this column is sucrose and this is cellulose, with the teal squares being low expression and the yellow squares being high expression. And as you can see, a lot of these PTH11 like GPCRs have high expression on cellulose, which could indicate that they are important for the breakdown of cellulose. And additionally, a previous study from our lab published by former grad student Ilva Cabrera in 2015 showed that 3 of these mutants, once you deleted the gene, had a growth defect when grown on cellulose. So having all this data together, our question is really, do these PTH11 like GPCRs affect cellulose degradation in Neurospora crassa? So in order to test this, we grew each mutant in liquid cultures that contain different carbon sources. In this case, it was glucose and cellulose. Once they grew, we collected the cell pads, and the total protein from the cellulose growth was normalized to the glucose growth. The reason we did this normalization was to rule out strains that just had poor general growth. We really wanted strains that had a phenotype specific to cellulose and ones that weren't just bad growers overall. So with these experiments, we found that 15 out of the 27 PTH11 like GPCRs that were tested had a phenotype. Of those 15, 10 of them had a reduction, which are shown with the red bars with the minus signs overhead, and 5 had an increase, which are the green bars with the plus signs overhead. The asterisk corresponds to level of significance, with 1 asterisk being a p-value of 0.05, 2 asterisks being a p-value of 0.01, and 3 asterisks being a p-value of 0.001. Now, I'd like to point out that it's pretty striking that of over 27 mutants, over half of them had a phenotype that we saw. Additionally, the ones that have phenotypes, so like as you can see here, these 3, 5189, 5307, and 5829, are all showed a reduction in growth, and they're additionally closely related to each other on the phylogenetic tree. They're all in the same clade. Furthermore, that previous study that I mentioned by Ilva Cabrera, of the 3 mutants that had a growth rate defect, 2 of the 3 mutants also had a growth rate defect from our data. So really what this data is showing us as to why they're not all just one phenotype and it's all just like decreased growth or increased growth. The reason for that being is that these GPCRs can have same ligands, but have different effects. And this can lead to like antagonism between the GPCRs, which can explain as to why some of these GPCRs are positive regulators. So the ones that have decreased growth, when you delete them, are likely positive, and inverse for the increased growth. So moving on to what we want to do with this data now that we have it, like what's next? So first, we want to begin creating these gene deletion multi-mutants. So that starts with the GPCR double deletion. We want to do this in order to find synergistic effects. So we want to like boost the effects of each other. And we're beginning with ones that have similar phenotypes and close relations. We've already started crossing some GPCRs that are particularly of interest to us. Additionally, we want to test for physical interactions between the G proteins and the PTH11 like GPCRs. Again, this has already started from one of the grad students in our lab. And finally, I'd just like to take some time to acknowledge everyone who helped with this. So I'd like to begin with Dr. Catherine Borkovich for allowing me to conduct research in the lab. I'd additionally like to thank Logan Collier, one of the grad students in the lab, for mentoring me through all of this. I'd also like to thank Yagna Oza, another grad student, for helping run the experiments. And finally, I'd like to thank NARCU-STAR and the NIH for providing the funding for this research. Thank you.
Awesome, thanks, Abel.